Hello, everyone. Welcome to Lunch Break. I'm Ariel Johnson from the Leakey Foundation. Oh, I Today we explore what fossil leaves and other botanical remains tell us about ancient primate ecosystems and more. Leakey Foundation Baldwin and Frank Brown scholar Venanzio Munyaka joins us from Texas in the United States, where he is a PhD student at Baylor University. Venanzio works in the field at the site of Koru in western Kenya, which is a Miocene site with some pretty spectacular fossils, including some fossil leaves, which we will have a chance to look at later. Now we'll just get to see this really cool site. So let's give a lunch break science welcome to Venanzio Munyaka.
Hey, thanks for having me.
Well, we're delighted to chat fossil leaves today. So you are a paleobotanist. What
I
exactly
am.
is paleobotany?
So that's a really cool question, and it's good we first actually understand what paleobotany is before we dive into the whole science of what it helps us with. Paleobotany is basically studying the evolution of plants, looking at old collections of plants, such as what we're seeing here is one of the famous botanical collection. It's called a lepidodendron, and it's a carboniferous tree. So we tend to understand what type of trees or plants are growing at these specific time periods, and if we would look at the time frame on how plants are evolving, for example, when we start seeing land plants coming up, to seeing vascular plants taking over, to arborescence, until the colonization of the whole ecosystem. So just having an idea of the evolution of plant groups. And another reason why we would study paleobotany is to understand the form and the function of these plants. For example, looking at when the leaves came up from pretty spiny things to like the basic leaves that we see, to evolution of roots where you're seeing like shallow roots, to these deep roots that we see on these big plants. And this form and function, which is in the evolution of plant traits, is one of the important reasons on why we study, what paleobotany is. Yeah.
So fossil leaves are just incredibly beautiful. We have a couple of images of some of them,
Mhm.
but there is more to them than meets the eye. Why are we interested in studying botanical fossils?
Yeah. Leaves happen to be one of my favorite paleobotanical collections, and why the reason why we actually love studying them is we understand that truly what we are seeing are actual plants. So we can say, like, for example, this is a collection from Koru 16. We can tell, like, yeah, there were plants, and these plants had leaves that look like this. So it's a direct explanation of what is growing in the past. Another important reason on why we study this is that there's this process on when we have the whole Pangea coming together. Wegener was able to see some of these plants and correlated the distribution of these plants on different subcontinents and put together the whole Pangea. So paleobotany is actually useful to understand the continental drift model. Another good reason why we would study this is on biostratigraphy, where we've seen multiple studies, especially ones that look at the first appearance of species to the last appearance of species. So pretty much like what we do with other paleontological collections, such as the inverts, where you see ammonites coming up and then you have an extinction event. So the same thing with plants is where you see like a big, like on this study they were looking at when there was the Chicxulub impact on 66 million years ago. So trying to reference yourself stratigraphically where you are in reference to the material that you're seeing. And the last point, and it's not the least one, but one that I tend to use a lot, is when we're trying to understand the paleoclimate. So this, when there's a correlation that has been developed between plants, specifically leaves, in their response to, for example, mean annual temperature, where we have this correlation between leaves that have teeth on them. So the leaves that you're seeing on the bottom side, so it has teeth, to leaves that are entire, so those that don't have teeth in them. So we see this broad distribution, and the correlation between this has been established to correlation of these leaves with mean annual temperature. Together with that, when we are trying to understand mean annual precipitation, is that we look at the different sizes of leaves, and this correlation has been established as well, and has been used to understand modern flora on how it responds to regional climate, and that translated to what's happening in the past in reference to paleoclimate.
Well, thank you for that introduction to paleobotany and why we look at leaves. Before we dig deeper, though, if you want to learn more about amazing discoveries about our distant ancestors or closest living relatives, as a non-human primates or human primates, hit the subscribe button and set notifications so you never miss a discovery. Now let's turn the virtual floor over to Venanzio and explore what primate fossils tell us about the climate, vegetation, and early ape evolution in the early Miocene.
Yeah. Thank you everyone for tuning in on this, and I'm gonna talk about this project that I have been working on it for the past 2-3 years, looking at the paleoclimate and the paleoecological context of an early Miocene habitat in East Africa. So since we will be talking about paleoclimate over and over, so I felt like it's good we understand what's pretty much happening during this time. So the evolution of these early apes is set on this background of a global cooling trend shown by this pink block right here. So that's the time period that I'm interested in. So there's this global cooling coming up from Oligocene heating up to the Miocene. And when you see these biogeographical changes associated with rifting, which happened pretty much in the early Miocene, which could have contributed to the climate and at par to the vegetation that we're seeing during this time period. More specifically, if we look at the vegetation itself, is that we're seeing this habitat heterogeneity from like close forests to open grasslands. And specifically, since I tend to work with phytoliths as well, is that this paper showed the distribution of these C4 grasses on different early Miocene sites, proving this point on habitat heterogeneity on these site-specific studies that are able to explore what's happening during this time period. But if we look at the early Miocene, is that we see that in many cases, quantitative and nuanced paleoclimate and paleovegetational reconstruction of these sites is quite limited, and we don't tend to understand pretty well what's happening in this time period of early Miocene. And this limits our scientific testing of the hypothesis that relates to the local changes in climate and or vegetation or on hominoid evolution. So that brings me to the main question, or what drives this whole study. So it's like, how did the early Miocene climate and plant community structure influence the evolution of hominoids? And the best place to do this is East Africa, where you have a good collection of these catarrhine and apes coming up from, early Miocene all the way to mid-Miocene. So where this site specifically, where you're doing this site-specific study that contains most of these apes, looking at the specific question on the paleo-environment and the paleoclimate of this site during the early Miocene. So specifically, or like putting everyone into geographical context, is that we are somewhere around here where you're seeing the East African Rift System and the associated Nyanza Rift. And on the top right, the panel C is the whole edifice of the Tinderet volcano. And we're seeing a site there called Songhor, which has been effectively sampled, but the paleo environment is not well understood. And when I'm talking about this project, I'm talking about this locality, Koru 16. And in that locality, we have two sub-localities. We have sub-locality A and sub-locality B. And between these two sub-localities will be this surface collection on the paleontological stuff. And when I'll be talking about quarry 1 and quarry 2, is that's what we're seeing on panel E, is these two quarries, which are about 5 meters away from each other, but they are all located in sub-locality A. So if we look at the stuff that has been collected from this site, so the fauna, is pretty much everything that we see in Core 16. You can find it in Songhor or other sites around the whole edifice itself, where you have about 55% of rodents, so these specific guys, and we've got about 21% of the primates. And more specifically, it's that we have this large Nyanza Pithecine, which is a highly successful group from the late Oligocene to the middle Miocene. Still try to understand why we can only see this large Nyanza Pithecine around this area. And of course, we've got a single taxon of Pythanoid, which one of the collaborators is working on the paleoecology of these reptiles, which, as I'm going to show later on, the numbers that we're getting on the paleoclimate relates to the reptiles that we are seeing from this specific site. So the geology of this specific site is basically this subaerial with fluvial influence of, we've got some weakly developed paleosols, we've got some ash layers in it, and specifically on this unit 4, that's where we did all the leaf collections from. But first, I'll talk about unit 6, where it has those trunk tree stumps. So those tree stumps, we use them to also understand the ecology. But first, we did by trenching the whole section, we collected these different paleosols and classified them to be pretty much inceptisols and vertisols. And these inceptisols were had a lot of mottling in them. We've got a lot of iron and manganese depleted zones, which we interpreted to be saturated conditions. And the vertisols have these slickened sides, which results from occasional swelling and contracting. So where you have these pressure surfaces occurring, especially when it's wet, so the soil expands, and when it gets dry, the soil contracts, and that's the slickened side. And we've got these angular blocky beds. So that's pretty much what you're seeing in unit 1. And this, as I had said, the paleosols are poorly developed, is that what we're seeing on that panel down there is we're seeing all these weatherable minerals on these specific paleosols. So remember, I talked about this unit 6 on this tree stump layer. So what we did with that layer is that we spatially plotted all these stumps. So you take each stump, take the radius, and then map it on a big region of about 600 meters wide. So plotting those stumps. So the first the top panel shows sublocality A, and the bottom panel is showing you sublocality B. And with that, we can use some allometric studies on like the tree base per height. So plotting it to about 1.3 meters and understand what type of a forest we are looking at. And on those on that specific layer, we also sampled the paleosol associated with that layer, and we found some microcharcoal present. And from this, we can interpret that we don't have a lot of big trees. And from that, it's plotting to something close to a tropical seasonally dry forest that has potential disturbance by the evidence of the microcharcoal from that layer. But then this is the stuff that I have spent a lot of time looking at. These are the leaves that we sampled from this layer. Remember when I talked about the good thing about paleobotany is that you go back to 20 million years ago and you can confidently say that, yes, you've got plants. And the first thing that a paleobotanist would see when they see such a collection is that you've got these huge leaves and you've got some leaves which are we call them their compound, such as what we're seeing on the bottom left corner. So these two leaves tend to be connected to each other. So it means they haven't been transported very far. They're pretty close to each other. And the fact we haven't taxonomically identified these leaves, but what we do is that we assign them to different morphotypes. So some of the figures you'll be seeing the word morphotype. So a morphotype is a word that is used to specifically put it in this species category, calling morphotype 1 to be a certain species, morphotype 2 all the way to all the morphotypes that we are able to collect. So we collect about 1,500 sensors on these leaves. And we used a variety of methods to try and understand what's happening to this whole flora. So the first thing we do, since most of these leaves are pretty fragmented, is that we try and reconstruct the leaf area. So what happens is that you take a leaf, as much as the whole leaf is not well preserved, we can measure the small boxes as shown in that green figure with those black boxes right there. Is that we use a method called leaf density measurement, where you take a leaf with all the veins coming off of it. So how we know is that when you have one leaf and you have the primary vein, which runs from the top to the bottom, all the veins connecting to it, that's what we call the secondary venation. So we take the secondary venation and we calculate small areas along those. So you take at least two successive secondary venations, and with that we extrapolate it with that transfer function shown by Saki et al. 2012, and we can get the area. When you get the area, then we can translate, as I had talked before, the relationship between leaf area and mean annual precipitation to understand the mean annual precipitation. And then you take a leaf and with the stalk in it. So when I talk about the stalk, I have shown you on the previous picture, is that thing that connects the plant to the whole branch. So that stalk, that's what we call a petiole. There's a relationship between that petiole with the leaf itself, and that's what we use to understand the leaf habit. So the leaf habit is, are you looking at a deciduous taxa or are you looking at an evergreen taxa? And lastly is that with the leaf itself, you can look at the whole margin of the leaf. So remember when I had shown you that previous curve with the relationship between the toothed leaf and untoothed leaves. So when you look at that margin, leaf margin analysis, we understand mean annual temperature. So the first thing, as any paleontologist will do, is first understand how much or how effective they were able to sample their site. So remember when I talk about quarry 1 and quarry 2, is these two 5-meter spaced quarries at sublocality A, and it reflects heterogeneity. So between these two spatially distant quarries, is that they've got about 55% of morphotypes shared between these two. And we've got a low species richness in this site, and the ecological diversity is not that huge, which is not surprising, since previous studies have shown that most of the tropical forests don't have that much of diversity in them. Then the next thing is taking all the leaves and distributing them between the smallest leaf to the biggest leaf to see how much large leaves you have, how much small leaves you have, instead of just like qualitatively looking at the leaves and being like, oh yeah, that's big. So there's a web class for classifying these leaves. And that's what we did with this plot here, showing that the line that is coming all the way, the green line, is the median of all these leaves. And it's what we can conclude is that we've got really large leaves from this particular site. So the climate, what we're seeing with the climate, we did three different regressive, we used three different regressive equations to just make sure that we are able to ascertain that what we're seeing is right. And we see a good overlap between the three of them to a really wet ecosystem. And the good thing about leaves is that we can like quantify that number. And that's what you're seeing on this box here, the bottom right one, where you're seeing Koru 16 has about 2 meters of rainfall. And using the leaf margin analysis is that it's pretty warm. We're getting about 24 degrees Celsius. And when you take those two variables, we can plot them on a Whittaker biome plot, which distributes in reference to like modern ecology, and see where Koru 16 plots. And it plots comfortably on a tropical seasonal forest. So the next thing that we did is that how I had talked about the leaf habit, the relationship between this big leaf with the stalk that it connects to it, is that we got some global data sets. So big shout out to Loie. He gave us this global set data set, and what we did is that we filtered it on these specific classes of reconstructed leaf areas. So taking a modern leaf and treating it as a fossil leaf, so reconstructing its leaf mass bar area, and then dividing it between deciduous, semi-deciduous to evergreen. And the gray bar that you're seeing crossing all the way from left to right, that's the transition between an evergreen and a deciduous taxa, which for tropical forests, sometimes it's not as easy, like a dichotomy, like just splitting. It's like we have that gray area of like, you don't move straight from a tropical deciduous forest to a tropical evergreen forest. That transition is sometimes not as smooth as it. So that gray line counts at it at about 129. And so using that modern taxa, we introduced all the morphotypes, so the species level, so looking at all the individual morphotypes from this area, the leaf mass bar area for those individual morphotypes, and then looking at it in a site level, so looking at the whole flora and seeing what we're seeing from it. And from this is that we have this two-tailed kind of representation. We have a number of good leaves as deciduous taxa and a good number of evergreen taxa. And since there's like so much coming on these leaves, we can take that and plot it on different modern distributions, such as shown on this figure, where we have tropical rainforest shown as a Kakamega forest. So this is a modern forest, and a tropical season forest such as one in Kibwezi, and a woodland such as that in Dar es Salaam, and then comparing it to the distribution of Koru 16. And if you superimpose this with Koru 16, is that we see that, two-tail where you have some deciduous and some evergreen taxa showing a distribution similar to a tropical rainforest to a tropical seasonal forest. So putting everything together in reference to this paleobotany, I mean, to the leaves that we sampled, is that the paleoclimate is pretty warm and wet climate with seasonal rainfall. The ecology is you have this mixed deciduous evergreen taxa, but we have mostly the evergreen taxa taken up. And then the whole biome reconstruct to a tropical rainforest to a tropical seasonal forest. The whole ecology showing this close canopy forest dominated by pretty much evergreen taxa. And that's pretty much what we see in Kibale National Park in Uganda, or this highland tropical rainforest in Cameroon, so the Banyang forest. So taking that and now placing Koru 16 into the other fossil sites in East Africa, is that Koru 16 now falls into this tropical seasonal forest to a to a closed it falls to this tropical seasonal forest with also a closed canopy effect in it. So I use two proxies to pretty much understand what's going on in paleoclimate and paleoecology. And then I looked at different early Miocene sites and middle Miocene sites and see what story is going on around this time period. And also bringing up the importance of these site-specific studies that look at this specific, ape where apes are, to try and constrain what type of habitats they are. So here I show paleosols and fossil leaves, and we see Koru 16 being one of these, end members, being a wet tropical forest, which the rate of sedimentation and accumulation is greater than how much the soil is able to form. That's why we are able to see these weakly developed paleosols that also have some fluvial effect on them. So also being to I mean, putting the stress on the seasonality on it by seeing those silicon sites on the paleosols. And lastly, connecting Koru 16 apes to this closed habitat environment, which underscores this importance of this broad distribution of early apes. And with that, I would love to thank all my funding sources, all the guys who've been working on this site from 2015, me picking it up on since 2022, the Leakey Foundation for all the help and the grants that they have provided me, and Baylor University, where I am right now, and my department, and pretty much everyone in my family. So thank you.
Thank you so much. That was an amazing look into your work. If you enjoyed this talk, please let us know by liking this video and sharing it. You play an incredible role in helping us share this amazing information. So please, again, like and subscribe. I have a question for you. So why should we all know about the site of Koru? I know it is the location of a very famous fossil. Do you want to talk a little bit about which fossil was discovered there and what makes that particular site such an interesting place?
So, Koru, we have this. We have a proconsul from Koru 16, and so this site taken together with other sites being studied by the larger group, the REACH. So it's a research on the early Catarrhine and hominoid evolution, where we try and understand the fundamental adaptive shifts of this body position, locomotion, the diets of these particular apes, trying to understand their origin and diversification of these hominoids, which also translates to coming all the way to the hominids. So when we understand this adaptive significance of this characteristic ape, is if we place them or if we study them in their context, so like Core 16 being the best context for this particular ape, we have a better understanding on what is happening to this particular clade or what makes apes apes. And then this can also be translated, or like when we're trying to understand what makes humans, then we have like a good understanding of this, and not merely those in which they were, later used at. So yeah, that's why I think Core 16 is it's a really good site that you have these plants and you have these apes being in the same kind of environment. So putting them right into their own specific context.
Well, we want to start taking some questions from our viewers. So if you have not gotten your questions in yet, please be sure to get those questions in now. We also want to take a moment to highlight the Francis H. Brown African Scholarship Fund. This fund plays an incredibly important role in expanding human evolution, knowledge of earth sciences and botany, specifically in relation to human origins. And by supporting talented East African researchers and other students like our guest today, Venanzio Munyaka, you can make more research, more groundbreaking research, and empower the next generation of scientists in East Africa by donating today. 100% of donations go directly to the program. Every contribution makes a difference. Let's make a lasting impact together. You can donate either through the link we shared in the chat or through YouTube giving. So, like I said, you now can submit any questions you have into the chat. And next week or not next week, I'm thinking back to when we had weekly episodes, but next month on Lunch Break Science, we'll be featuring Cheryl Knott. who will be discussing orangutan conservation. So let us take our first question from our viewers. Rahab asks, what altitude would you place Koru 16 site during the Miocene? Is it a similar altitude as present day or different?
Yeah, that's a really good question. Koru 16, the deposition of Koru 16 itself, it's prior to and during local rifting of that Nyanza Rift. And this would mean, well, we haven't well studied the altitude of this site, but we assume that there isn't such, a lot of elevation change between this. So this happens that this site would come to about, I'm trying to get the numbers right, comes to about 2,300 meters. So that's the modern elevation right now. And elevation is one of the major drivers to flora changes, as much as that we have differential in soils. We've got this. So look at this. So when you have this elevation change, you tend to create these micro habitats that would have resulted to these specific ecosystems developing from those. So there's definitely a big influence on elevation, the elevation of Core 16 to what we're seeing with ecology of that site.
So you don't just work at Koru 16, you also work at many other sites. What are some of the other sites you work at?
Yeah, so since this is my first part of the dissertation, the second part I work in a Miocene site in Sondu. So this is a different formation and also younger than this site. So what I do still, I'm working on paleosols, and I'm using a different proxy called called Phytoliths, and these are biogenic silica produced by plants. So as plants are taking up water and transpires, is that they deposit the silica, which goes into the whole plant, and they get really well preserved. And actually, we've seen like a good case study on use of these phytoliths on a recent paper looking at the evolution of C4, seeing like, oh, maybe we can push C4 to earlier than we thought. Like we're able to see this specific biogenic silica as early as the Miocene of East Africa. So what I'm doing, I'm just revisiting the Middle Miocene to see what's happening. So this site is called it's in Sondu, so that's part of Western Kenya. So we've got different exposures, such as one in Chepete, we've got Cameroon, and just trying to compare what these two sites mean ecologically, and also employing different methods such as isotopes and complementing what these phytoliths are showing with it. And also, I've been working at a later site. I've been invited to work with Dr. Kinyanjui at the National Museum at a site in Lemudongo. This is a 6 million old site still. That trench. Looking at also the geology of this, sampling the tephras and seeing the changes in ecologies, still using phytoliths on those. And also, there's also another project in Western Turkana, which I work with my advisor, and also Dr. Lukens and Professor Susie Cody, well, with also a bunch of great undergrads who come with us looking at still paleoenvironmental contexts on this early Miocene site. So yeah, a lot of work going on in East Africa.
And you don't just look, I mean, the fossil leaves you showed us, and they're just spectacular, are quite large, but you also look at leaves that are much smaller, or botanical remains that are much smaller. Do you want to tell us a little bit about what the smallest remains you look at are?
Yeah, so yeah, those are the phytoliths. That's the plant silica, and they're really, really small. They would range from, so like shown on this, they range from negative, I mean, sorry, from 5 to about 200 micrometers. And yeah, the best thing about these guys is that since grasses are not the best preserved, they're not well preserved as how we are seeing these dicot leaves, as I had shown on the previous slide. These phytoliths are able to ameliorate that problem. Is that you just sample that paleosoil, you go do all the cool chemistry with it, and then you get stuff like this that we can be able to actually categorize what we're seeing. So they're good when you're trying to understand how close or how open your ecosystem is. So they give you that signature of the grasses that you might be missing in your floral record. And as I had said before, they've pretty much done a good job to like show that we actually have had C4 grasses in the early Miocene, and most of the research that has been done specifically in East Africa on the phytolith work has been from about 10 million years going all the way forward. So yeah, these are one of my favorites. And actually, when I started paleobotany, this was my first proxy to work with. That's why yeah, it's pretty nice. Yeah.
So we have a follow-up question from
Mhm,
H. Gregory.
mhm.
Um, how do fossil leaves compare to phytoliths when reconstructing past environments? And also, like, I'm also curious to build on that question. Like, how frequently do you find each type?
Yeah, so this is they complement each other really well. So the one the disadvantage of like working on Koru 16 is that the geology of this place is a carbonatite. It messes up everything and it messes up the geology. In an ideal world, you would sample these paleosols where you have these good preserved leaves, and then you collect these paleosols from these specific sections, and you use some vegetation indexes, especially on phytoliths, and you would look at forest indicators, and you look at grass indicators, and compare the aridity indices on this stuff, and then compare these two where you have the leaves telling you that, hey, you have this kind of ecology and you have this much of precipitation, and then the phytoliths come in and tell you, yeah, you have, so this is how your whole vegetation looks like in reference of like how open or closed it is with it. So they complement each other really well. It's only that for example, for this side, because I also tried to extract the phytoliths from Koru 16, and what you pretty much get on these volcanic deposits is just silica, which is just volcanic glass. And since, well, the densities between these two are different, so we've tried to modify the density to see if we can just have the biogenic silica in the process of extraction to see if we can get a good record off of it. But yeah, unfortunately, it's not working. But they work in hand, and most of the time they get to agree with each other.
So our next question comes from Berryville Cat. Were you able to determine plant species from some macrofossils? Were they fruit-producing species?
Yeah, so yeah, we haven't clearly established the we haven't taxonomically identified these leaves, but some of the leaves there we can base them off of like a one of the leaves. I know there was like a strychnose in there. We've got some palms, we've got some fruits such as this guy, and we're still trying to determine where or like who is this guy. So yeah, we've got potentially some of the morphotypes that were fruit bearing. Yeah, so still that's stuff that is under review right now, trying to constrain this these two, the taxonomy and the kind of fruiting bodies that we're getting from this particular site. Yeah, good question.
So you are part of an amazing team out in the field, but it's not just excavating fossils and scientific research. You're a close-knit group. What is your
Right.
team like, and what else do you get up to in the field?
Yeah, yeah. When you're working in these sites, yeah, you need to have like a good and a loving group, and like having great people and a great team to be working with. And I greatly appreciate everyone who comes out with me every time that I go out to do a dig. And I know, and it's always crazy when we are like digging out there, is because, yeah, like this is a good example of one of the quarries is that we dug that quarry one day ago, and we exposed that leaf layer, and then it stormed that previous day. So that previous night. So when we're coming the following day, the whole quarry is full of water. You have guys who are here with you, and they're helping you dig and do everything. And if you don't have a good relationship and you're telling everyone, hey, we need to like bucket this water out and then leave it to dry and then keep digging. So it's a lot of friendship, and I don't know how much I can appreciate everyone who comes out with us. Stuff like here is one of my birthdays. It happened that I was still in the field, and I got surprised by the cook there. He made this wonderful cake, and it's I sometimes I even lack words on how great friendship, or like there's no disparity between, oh, being like you're a student versus a researcher. Or like sometimes we have Father's Day and we have to make a dance, sometimes cut a cake to celebrate this. So there's so much love, there's so much science, there's a lot going on, that for like the 3 weeks or 4 weeks that we are out in the field, you, as much as you miss being at home, you have this home in the field, and you're like, oh, as much as everyone is a professor or a scientist, they're fun guys to hang out with. So yeah. Mm,
It sounds like a great group to be with.
yeah.
Um, we have another question from Two Faces Quantum Figures. The question is a little bit longer, but what does the environment specifically tell us about proconsul? Is what I would sum this question about.
Yeah, so specifically, since I heavily don't work on the proconsul or the paleontology of this site. So my responsibility is pretty much telling them the context on the evolution of this stuff. So I think the question that comes from like having this paleoecological construction is that what was like the distribution of this particular ape? We put that into question, being like, was it like a closed forest, or was it an open forest? And that's why I also like brought that need of specifically like going to these site specific studies that try and reconstruct the paleo environment on these particular apes. And for example, like the study that was done in Uganda on Morotopithecus, is that we're able to understand what's happening to that particular primate by also knowing that, oh, it had this kind of a diet. So some of this stuff you could understand by if you reconstruct the whole ecology. So I might not be able to answer what this would mean for this particular clade proconsul. My only point that I will bring is that maybe there's a big variation, or like it puts into question the habitats that this particular proconsul lived in could have been this particular forest.
So, how did you first become interested in studying paleoanthropology, and what has your journey been like?
Oh yeah, so a big shout out goes to Rahab, Rahab Kinyanjui. She's an amazing researcher, and she pretty much, I would say, found me stranded somewhere in the field of science. This was when I was still in college, still trying to decide what I'm gonna do. And few months before I graduated, she invited me out to go and do some field work with her. And also give a big shout out to Dr. Tamo, and he was looking at developing this proxy on understanding different distribution of leaves in these low latitude sites and what that means in relation to ecology, I mean, in relation to climate. So that's the first time I went out to the field and we sampled all these leaves, and he put me up on like doing we call it clamp, so doing all these trades and developing this whole data set, which I didn't even know what's gonna happen with it. And then after that, then after we kept talking, and oh yeah, that's me back in 2018, somewhere in a glade in Kakamega forest. And from that time, that's when I grew the interest on like, oh, so there's more into these plants than what we just see them, and we can use these plants to understand the ecology, to understand the climate and everything. And that's how I came all the way and got to meet Dan Pepe, and he had this amazing project, and I was like, yeah, I'm new into it. I just started learning about these modern collections. So, and the whole conversation about Koru began from there, and yeah, here we are now.
So, you gave a couple of presentations this year at various societies. How has it been being out there, sharing your work with your peers?
Yeah, as an African student, going out and presenting on these big podiums and everything, the first time it's like, oh, I don't know if I can do this. It's wild. It's crazy. That's a few months ago during EAPP, and this is somewhere in Michigan. Still talking about the first results of these projects. It's always a great opportunity for you know students like me going out and presenting your science and getting all this uh, constructive criticism from everyone, being like, hey, I think you can go and modify this. Hey, I think you should look at this. So it's always a good podium to be able to show what you're doing, introduce people to paleobotany, such as what we're talking about right now, and show them like, oh, still in East Africa we've got all this cool stuff that is going on. And yeah, sharing science with everybody. And yeah, it's always a great, and also mentoring few students in the field and stuff like that, like whoever, like every other student who comes with me out in the field to help with field work. It's always a great pleasure to share whatever I know with uh fellow Kenyans back at home. Yeah. Mm.
Well, it's wonderful to hear about your story and your background.
Mhm.
Is there any site you would just love to work at that you've not worked at yet?
Oh, oh boy.
Totally
Not
putting you
yet.
on the spot.
Oh yeah, well, there's just so much. Like I had shown you on that on my presentation on the importance of these specific site specific studies. I would love to like go back to these early Miocene sites, like some of the proxies that have been used before. Right now they have been improved. So like revisiting these sites and applying new methods on those sites and trying to constrain what's happening on, in reference to paleoclimate on this side. And also with that question that was brought about, being like, we have this more the relationship between the paleobotany, I mean the leaves and the phytoliths, is like putting these two proxies together and seeing what these sites can tell us. So yeah, ideally there's a good point that I would think about on the spot.
So, do you have any advice for those who are watching out there who are thinking of pursuing a career in science or early in their careers?
Yes. I would say first, you have to, respect your mentors and be quick to listen. Um, this is sometimes a difficult science, sometimes, uh, and especially if you're like an international student having to, like the first figure, I mean, the first image which you showed, like me being in Baylor, and then you had to travel all the way 8,000 miles, being like, sometimes it feels good being that far that you get to meet all the people doing good science everywhere. But all I would say is that humble, being ready to learn, again, being quick to listen, uh, and just follow your dreams. I think anything is achievable. Like I didn't know about paleobotany until like I got to graduate from my college, but it's more like just being ready to learn and just being ready to learn. Yeah, that's the main point I would say. Yeah. Oh, yeah, that's a great figure of back in 2019 when I got to participate in Kobe for a Fit School. Actually, a big shout out to KFFS. This was my first actual field work, like going out and spending 6 weeks doing crazy stuff. Yeah, this was my that learning curve, getting introduced to paleo world. Yeah. Mhm.
Thank you so much. Thank you for joining us today. This was just a spectacular look into ancient ecosystems, and we're just so happy to have you here.
Yeah, absolutely. Thank you so much.
We'd also like to give a special shout out to our live viewers, Sanyatan, Nilius, Pete, Two Faces, Deepak, Berryville Cat, Vale, Rahab, H, Sears James, Beth, and everyone else who's watching who hasn't made a comment. We're just delighted to have you, and thank you so much for tuning in. And be sure to share this episode if you enjoyed it so that we can, share Venanzio's work with more people.
Thank you.
Well, thank you all for taking a break from your day and feeding your brain with the Leaky Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leaky Foundation and made possible by the generous support of the Ann and Gordon Getty Foundation, Camilla and George Smith, and the Joan and Arnold Travis Education Fund, as well as viewers like you. Show your support of Lunch Break Science by subscribing to our channel, clicking on notifications, and giving us a thumbs up, or making a donation to help us create new content. Still craving science and can't wait for the next episode? You can feast on the Leaky Foundation's content library with past episodes, lectures, our podcast origin stories, and more. Thank you all for tuning in, and see you next time.